So today we're going to be learning about active transport. And if you remember from the past few days, you've been discussing a lot of passive transport. And if you recall, passive transport is the one that requires no energy. So today we're going to talk about active transport, which, if you can guess, does require some energy. So just as an overview, active transport can be divided into three separate categories. The first one we're going to talk about is when we use a carrier protein. The second one that we're going to discuss is endocytosis, and finally exocytosis. So remember, these are all still ones that require energy. So they do require energy to make these happen. So overall, active transport, the first one we're going to talk about is when it has to do with a carrier protein. And if you remember, we did talk about proteins in passive transport, but the proteins in those situations were always going from high concentration to low concentration. But if you notice here, carrier proteins and active transport have to do when we are moving molecules against the concentration gradient. Okay, and when we go against the concentration gradient, we are going from where there is not a lot of stuff to where there's a lot of stuff. So remember, this is kind of going against the grain, okay? Our body doesn't normally like to do this, but in some cases we need to. So anytime you are going from an area of low concentration to an area of high concentration, it is going to require some energy. You can see energy is required to make that happen. So what it does is it uses ATP, and ATP, if you remember, is our energy source, okay? So it's going to use the ATP to flip over and move the molecule to another side of the membrane. So if you look here, this is the area of right here. So we have a low concentration here, and here we have a high concentration of ions. Okay, so what we're trying to do is we're trying to get the molecule to go from this area of low concentration, and we want to get these molecules in here. So we're going against what's normal. Okay, normally you can see that there are lots of molecules down here, and there's not a lot of molecules up here. So the normal pathway would be to go from here to here, here to here. Okay, but this is one that's trying to push it the other way. So in this instance, we are going to require some energy here. So this energy, okay, if you can't see, it's right here. So this energy is going to be required to make this process happen. Okay, to make those molecules move against the concentration gradient to get from where there's not a lot of stuff to where there's a lot of stuff. Okay, so what happens is that the ion will come into here, and it will come into the carrier protein, and you can see that it's blocked right here. And so the carrier protein will actually flip, so they kind of flip themselves, and they open up, and then this molecule can then be pushed in. Okay, and that does require some energy. So one example of this is the sodium-potassium pump, okay, and this is found in our bodies in many, many places along cell membranes, okay. We need this to be able to have this different concentration of sodium molecules. If you remember, sodium is Na, okay, and we also need it to do it for the potassium ions, which you can see here. Hopefully you remember potassium is K, okay, and they're called ions because they have a charge. So Na is positive, potassium is positive. Okay, so what this pump does is right here you can see that the sodium is attached to the side, and right now you can see that there's lots of sodium up here, not a lot of sodium up here. So to get these three sodium to actually go inside, it requires some energy. And if you can see that there, this is actually a molecule of ATP. Okay, ATP, if you remember, means adenosine triphosphate. So here are the 1, 2, 3 phosphate molecules. Okay, so it requires one of them. It breaks off. The potassium, or sorry, the phosphate stays with the molecule here, and then the 2 phosphates and the adenosine break off. And so that's what's happening here. The phosphate has stayed, and now what it's done is you can see that the pump here was closed at the top, open at the bottom. And now down here, it's open at the top and closed at the bottom. So it's literally kind of flipped itself and opened up so that now these 3 potassium can get pumped into this area. Or sorry, these 3 sodium. Same thing over here. Okay, there's not a lot of potassium up here, and there's lots of potassium in here. So this pump is trying to work against the concentration gradient. Okay, so it's going against the concentration gradient. It's trying to get in where there's already a lot of it. So what will happen is again, another phosphate here, so another molecule of ATP has come in and left that phosphate there, and it's given enough energy from where the bond was broken. And then the potassium here attach. Once that happens, the phosphate comes in, and then this molecule, you can see that it was open at the top, closed at the bottom. Now it's closed at the top, open at the bottom. So it's kind of flipped itself, and now the potassium can get into where there was already some, and now there's even more. Okay, so that's a common example of a carrier protein and how it works. Okay, oftentimes in our human body, you'll see them called pumps, and that means the same thing. You're trying to pump it against the gradient. Okay, the next type that we're going to talk about is bulk transport, and bulk just means that you're moving large molecules. Okay, so these would be things that either you have a lot of them, okay, so in this instance you can see that there's a lot here trying to get in or out, or it could be that they're actually very large molecules. Okay, so, So right here, first one we're going to talk about is called exocytosis, and exocytosis has to do with moving solutes or fluids. Sorry, that's cut off. That says out of the cell. Okay, so we've got our molecules here that we need to get out, okay, and you can see that they have their own phospholipid bilayer wrapped around them. Okay, the reason why is because we don't want these molecules to interact with whatever's in the cytosol. So perhaps there's some enzymes in there, whatever it is, they don't want these molecules here to just be free floating around. Okay, we need to get rid of them. So it could be wastes, it could be things that we don't require, it could be anything. So what they do is they get themselves close to the cell membrane here, and you can see they kind of interact with each other, they almost combine. Okay, and then it becomes one large cell membrane, and once it connects with it, then it can release all of these solutes out into the extracellular fluid. So the extracellular fluid is just the outside. So this is outside of the cell, and this is inside cell. Okay, so all of these molecules here that were inside the cell, they're going to mesh themselves with the actual cell membrane, and then they're going to get pushed out. Okay, so that's exocytosis, and I always think of that when I'm trying to remember, is I always want to think of exo. To me, I remember exit, and that helps me remember what the difference is between the two. Okay, so the next one is endocytosis. So I apologize, this slide is a little bit cramped in here, but endocytosis, okay, to me, whenever I'm trying to think of that, I always think of enter. Okay, enter. So this is when you were trying to get stuff inside of the cell. So in this case, we were trying to get this food particle inside, and this one actually comes in as a food vacuole. Okay, so there's two different types here. The first type that we're going to look at is called phagocytosis, and phagocytosis is right here, and it's when large particles, whole cells or solids, enter the cell, and they enter themselves as a food vacuole. So we did talk about vacuoles when we talked about bacteria and viruses and different things that take materials into themselves as vacuoles, and then they can use it later. Okay, other cells do that as well. So this food would enter, and because we're bringing in a lot at once, that's why it's called bulk, just like bulk burn. Okay, so you get lots of stuff at once, and this would then stay in as a food vacuole, and then this membrane would just break down whenever the cell needs it. Okay, so the first thing is a food vacuole. The second kind is called pinocytosis, and pinocytosis has to do with liquids. Okay, so oftentimes you'll see the difference between there's cell eating and cell drinking. Okay, some people will call it that. So whenever you're trying to bring in large food molecules, they come in as phagocytosis. Okay, when you're trying to bring in liquids or dissolved solutes, they come in as pinocytosis. Okay, and these things, again, I apologize, this word here, okay, is a vesicle. So this comes in as a phagocytosis comes in as a food vacuole, and the pinocytosis comes in as a vesicle. Okay, and the vesicle is the same concept. It just has the cell membrane around the outside, and then the liquid on the inside. Okay? So down here it says, in both processes, the cell membrane wraps around the material being brought into the cell. So you can see here that the cell membrane is actually wrapping itself around, and it will eventually close off, and then that thing that closes off comes in into the middle, and then the cell membrane just reforms itself. So same thing here. This was a normal cell membrane just right across here. What it does is it actually pinches off. Okay, this will eventually close up again, and then that vesicle goes inside. So the two concepts again, phagocytosis is bringing in food particles, pinocytosis is bringing in liquids. So this is just another picture of that. Here's your food particle coming in, here's your membrane. The plasma membrane, remember, is the same as the cell membrane. Okay, and the cell membrane will eventually wrap itself around, it pinches off closed, and then this food vacuole goes into the cell to be used whenever it's needed. Okay, so overall, this is an overview of the entire process. So first thing that we're going to look at here is passive transport. Okay, and passive transport, remember, requires no energy. Then over here we have active transport, and that's what we looked at today. And you can see that active transport does require energy. So I want you to look over here on the right, and you can see the concentration gradient. Okay, and this is important because you always want to talk about whether it's moving with the concentration gradient, okay, with or whether it's moving against the concentration gradient. So you always have to decide whether it's moving with or against, and we also call that up or down. Okay, if it's moving the way it's supposed to, if it's moving from where there's a high concentration down to a low concentration, okay, we call that it's moving with the concentration gradient, or it's moving down the concentration gradient. Okay, I always think of it as like downhill, right? Is it easier to walk up the hill, or is it easier to roll down the hill? Okay, so in this case we'd be going down, and that's with the concentration gradient. If you're moving against the concentration gradient, that means that you are going against what's normal. Okay, so you're trying to move from an area of low concentration up to where there's lots of stuff already. Okay, we also think of that as you're moving up the concentration gradient, meaning you're trying to walk up the hill versus rolling down it. Okay, so we have our simple diffusion. So if it's a small enough molecule, okay, it can just go right through the lipid bilayer. If it's one that is a bit larger, sometimes it will need a channel protein, okay, or if it doesn't want to interact with the lipid bilayer. Okay, so for instance, if it's a larger molecule like glucose, or potentially a molecule that doesn't interact well with the fatty acids in here. Okay, the third one is facilitated diffusion, so it's going to grab it and it's going to open up on the other end and then move in here. Okay, but the big thing with all of these is that they are all moving, they're all moving in this direction. They are all going from an area of high concentration to low concentration. Okay. Once we get into the active transport, which is over here, okay, you'll notice that this protein and this protein are both called carrier proteins. However, the big difference is that now we are traveling in this direction. Okay, we're trying to go from an area of low concentration to high concentration, and that requires some energy. Okay, so when you are trying to move in that direction, when you're trying to go from where there's not a lot of stuff to where there's lots of stuff, it does require energy to do that. And so the molecule will come in here, ATP will come in, and it will flip it, and then this molecule can get inside like that. Okay, so we're going to watch a little video here, and this is just an overview of everything you've learned in the last few days.
The plasma membrane regulates the passage of molecules into and out of the cell. It's capable of carrying out this function because it's selectively permeable, meaning that it allows certain substances to pass while preventing others. Basically, substances enter the cell in one of three ways: passive transport, active transport, or bulk transport. Let's look at each of these in turn. Passive transport is the movement of substances into or out of a cell without the expenditure of energy by the cell. One form of passive transport is diffusion. During diffusion, molecules move across a membrane from an area of high concentration to an area of low concentration. The molecules are therefore said to be moving down a concentration gradient. This continues until equilibrium is reached and the molecules are distributed equally. Another form of passive transport is facilitated diffusion. Facilitated diffusion occurs when an ion or molecule diffuses across a membrane faster than expected, either by way of a specific channel protein or with the assistance of carrier proteins that change shape as they pass through. The diffusion of water across a membrane, or osmosis, is another example of passive transport. In many cases, specialized proteins called aquaporins allow for the more rapid transport of water molecules. The second type of transport, active transport, requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as up or against the concentration gradient. One of the more common examples is the sodium-potassium pump, which moves sodium ions back out of the cell and potassium ions into the cell. Notice that the sodium-potassium pump undergoes a change in shape that allows it to combine alternately with sodium ions and potassium ions. The third type of transport, bulk transport, is used for molecules that are too large to be moved by transport proteins. Instead, vesicles take them into or out of the cell. During this process, the plasma membrane surrounds and engulfs the particle. This is known as endocytosis. Cells use three basic types of endocytosis, depending on the size and nature of the material to be digested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material taken in is large, such as bacteria or a food particle, the process is called phagocytosis. Pinocytosis occurs when vesicles form around a liquid or very small particles. During receptor-mediated endocytosis, molecules bind to specific receptor proteins embedded in a coated pit within the plasma membrane. When enough molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a vesicle. Exocytosis is the opposite of endocytosis. During exocytosis, membrane-bound vesicles move to the surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell. To recap, there are three main types of cell transport: passive transport, active transport, and bulk transport. Passive transport is the movement of substances in or out of a cell without the expenditure of energy by the cell. Active transport requires energy in the form of ATP to move molecules against their concentration gradient. Bulk transport requires vesicle formation and metabolic energy. Forms of bulk transport include endocytosis, pinocytosis, receptor-mediated endocytosis, and exocytosis.